Hello, everyone. I'd like to continue talking about some unique aspects of the sex chromosomes. So, what we've talked about previously is the idea that when a gene resides on a sex chromosome, whether it be an X or a Y, in those organisms that have that distinction between males and females using X and Y chromosomes, if you have a gene on these chromosomes, what we've seen is that they're inherited in a unique manner. So again, X-linked traits are inherited differently depending on whether you are a male or a female. And again, the reason for that is because of the fact that females have two copies of the X chromosome and therefore two copies of all of those genes, all of those sex-linked genes. And males are hemizygous. They only have one X chromosome and therefore only one copy of an X-linked or a sex-linked gene. So this gives rise to, the unique inheritance patterns that we've talked about already, whether it be for a sex-linked dominant or sex-linked recessive. Okay, it's the same thing for a Y-linked trait. So a gene, a holandric trait. Again, that's a gene or a trait that is determined by a gene for which males only will inherit one copy. It always comes from the father. And females, a normal female will never inherit any copies of these Y-linked genes. Okay, so but we talking about the nature of these unique inheritance patterns. There are other aspects of the sex chromosomes that are interesting as well. So the first of these is the idea that there is a dosage difference between males and females for sex-linked genes. Okay, so again, if you think about it for a second, for all of the autosomes, the 22 autosomes in the human genome, both males and females have two copies. Okay, well, that's also true for a human female for the sex chromosomes, for the X chromosome. She will have two X chromosomes, two copies of every gene that falls on that chromosome. But that's not true for a male. So again, the male will only have one copy. So what we will see in an upcoming lecture next week sometime is the idea is that if you deviate from that normal diploid condition, two copies of each gene, if you deviate from that normal diploid condition, that as for autosomal genes, is that generally causes some sort of a developmental disruption or developmental abnormality and give rise to a particular genetic condition. So in other words, diploidy seems to be the normal state or the normal condition in terms of the organization of the human genome and most life forms on the planet. Diploidy seems to be the most common um, uh, state. So having two copies of everything seems to be normal, but that's not true in humans for sex-linked genes. Again, males will only have one copy. So why is that normal? Why is that the normal condition for the male? So this is what we're going to talk about in this lecture. So in addition to that, in an upcoming lecture, um, we're also going to talk about another unique aspect of the sex chromosome. So this is the idea that if there is a difference between males and females, then presumably there's genetic information on these chromosomes that determine the sex of the individual. In other words, develops or contributes to a developmental program that produces male or female reproductive systems. So again, we'll explore that a little bit more in an upcoming lecture. But now I want to focus on this idea of a dosage difference and why that is tolerated, why that's okay, or why that's normal for humans and other mammals. Okay, so they began to address this question more directly in the late 1950s. So I've shown you depictions of human karyotypes already, many different examples of that. And the ability to create a good interpretable karyotype for the human Actually, that technology wasn't fully developed until the mid to late 1950s. So it wasn't until the mid to late 1950s that you could produce a karyotype and look for, you first of all identify all 46 chromosomes as being distinct from each other, and having definitive evidence for this sex chromosome difference between males and females. And then you can also use that, as we'll see in an upcoming lecture, to interpret chromosomal abnormalities, chromosome number abnormalities that are responsible for certain genetic disorders. Okay, so in any event, with that understanding, a clear technology that allowed for, clear recognition and distinction of X chromosomes and Y chromosomes, knowing that females are XX and males are XY. This is when this question of, the dosage difference began to be addressed uh, very directly. So the woman, a woman by the name of Mary Lyon, was the first one to consider how this would be a situation that would be tolerated. In other words, having this dosage difference between males and females. And what she proposed is something that is now referred to as the Lyon hypothesis. This is around 1961 or so. And so she came up with this idea of something she called dosage compensation. And so what she suggested is that one of the two X chromosomes in the female actually becomes inactivated, so that the female then is similar to the male, having only one functional X chromosome and one functional copy of each gene that falls on the X chromosome. So there's a little bit more to that theory or that hypothesis, this form of dosage compensation that Mary Lyon proposed. But where did she first, even come up with this idea that one of the two X chromosomes would be inactivated in a human or in a mammalian female? So this was work done by this individual, a scientist by the name of Murray Barr, M.L. Barr, and this is work that goes back to the 1940s. Okay, so what he did is, very simple work. He was simply looking at different types of mammals, and he would take, somatic tissues, and he would process those tissues, and he would stain them so he could observe, the nucleus of the cell. And he observed a consistent difference in terms of the presentation of the nuclei of these somatic cells, depending on whether he's looking at a mammalian female or a mammalian male. And so what he observed was the following. So here's a cell, two cells, I guess, that are stained for easy visualization of the nucleus of the cell. And you can see in this particular nucleus, there's this darkly staining body indicated by the red arrow. Okay, so this is a cell that's derived from a mammalian female. Okay, here's the same tissue processed in the same way. And again, when you look at the nucleus, readily identifiable nucleus, you don't see that darkly staining body. And so this was a consistent observation by M.L. Barr for somatic tissues from many different types of mammalian organisms, not just the cat, not just humans, but many other types of mammals as well. So he published this information. So maybe not a result, but it's a clear distinction that occurs between the nuclei of the somatic cells of the different sexes of many different types of mammals. So this was in the literature, and so Mary Lyon had access to this information. This darkly staining body eventually became referred to as a Barr body. And according to Mary Lyon's hypothesis about dosage compensation, what she suggested was that Barr body represented an inactivated X chromosome. So again, remember in the nucleus of the cell, these would be interphase. You can see chromatin. You can't distinguish individual chromosomes. Is that what she suggested is that inactivated X chromosome became highly condensed and highly packaged so that all of the genetic information was contained within it was shut down or turned off. So then within that female nucleus with the Barr body, there's only one functional X chromosome that is in that uncondensed state that would allow for, functionality of the genes. So just like a normal human male with one X chromosome. That would also explain why you don't see a Barr body in the nuclei of somatic tissues of mammalian males, because they do not have an inactivated form of the X chromosome. So this served as the basis for her hypothesis. There's a little bit more detail to it. So in addition to that, so here here's a schematic of what she proposed. So here's a female zygote, two X chromosomes, again, one maternally derived, one paternally derived. Okay, so what she suggested is that during very early development, that cell would again begin to propagate itself mitotically. Eventually, what would happen is that when we just reach, I just said it arbitrarily, say 1,000 cell stage, so a ball of cells, a blastula, say consisting of about 1,000 cells. What she said was that at that stage, at that very early developmental stage, that 1,000 cell stage, is that the X chromosome becomes converted into a Barr body in each of those 1,000 cells. Okay, so that's again being represented here, this very early stage of development when X inactivation actually takes place. Okay, she also suggested that when the Barr body forms is that it's randomly formed. In other words, again, remember, the maternal and paternally derived X chromosomes can exhibit genetic differences at, at the allele level. So there's a random inactivation of one of those two X chromosomes in each of these 1,000 cells. Okay, then what she suggested is that beyond that point is that once one of the two Barr bodies becomes inactivated through all subsequent mitotic cell divisions, the same X chromosome remains inactivated. Okay, so in this case, this cell here, this is the maternally derived X that is inactivated, and all subsequent daughter cells that are produced through subsequent mitotic cell divisions retain that same maternal X in an inactivated form. So what that would then suggest is that in the fully formed individual, whether as an infant or a fully grown adult, is that a female will be a mosaic or a chimera in terms of which X chromosome is functional in any cell within the body or groups of cells, tissues within the body. So again, here's a fully formed female, and you can see sectors of the body that retain the maternally derived X chromosome in an inactivated form, and then other sectors of the body, tissues and cells within the body that retain the paternally derived X chromosome in an inactivated form. So as an adult or as a fully formed individual, she's a mosaic or a chimera for which X chromosome is functional in any given region of the body. So given all of these different components to her model or her hypothesis about dosage compensation in mammals and humans, is there any direct evidence that supports this particular hypothesis? So of course there is, and we'll just use one of the best examples, textbook examples, is in the cat, coat color in the cat. So again, there's an O gene that is sex-linked, so it's on the X chromosome, and there's two allelic alternatives. There's orange, big O, and there's black, little o. or lowercase o. So what would you expect for the fur color, the coat color, to be for a male cat? Well, it's going to be either solid orange or solid black, because the males are, these, again, cats are mammals, so the males are XY, they're hemizygous. They're either going to be carrying an orange allele or they're going to be carrying a black allele. But what about a female? So again, a female can exhibit three different genotypes for those two alleles. You could be homozygous orange. If that were the case, it would be an orange-coated cat, female cat. You could be homozygous black, okay, and then of course you'd have a solid black coat of fur on the female. Or alternatively, you could be heterozygous. And if you are heterozygous, this is the phenotype that results: a tortoiseshell coloration pattern, sectors that are orange and sectors that are black. And again, that's because of the X inactivation that occurs at that very early developmental stage that Mary Lyon described. So what you also notice is that when you look at different tortoiseshell colored female cats, is that the pattern is never the same. And so what that argues for, here's another example. Here's a couple of other, here's another example here. Here's another example here. So the idea is the random nature of the mosaicism or the chimerism is the reflection of the random inactivation of the X chromosome that occurs at that early developmental stage. Okay, so the other thing I want to point out is, coat color determination, the genetic basis for coat color in cats, it's more than one gene. As we've talked about previously, it's almost never that you see a situation where a given trait is determined by a single gene. So there's other genes that contribute to coat color in cats. And just to give you an idea of something that reflects that, it's calico cats. So again, the calico cats are tricolored. So they're orange, black, and then they have sectors of white. Here's two examples. I guess this one has a little bit of white as well. So what causes the calico coloration pattern? It's because there's another gene in addition to this O gene on the X chromosome that acts epistatically to the O gene, and it's autosomally located. And what it can do is it can mask or modify expression of the O gene within the fur of the cat. So in order to produce the calico, the tricolored pattern, you have to be female, you have to be heterozygous for the O gene, and then you have to have this autosomal gene acting epistatically on that O gene. You have to have the proper allelic presence at that autosomal gene in order to produce that tricolored pattern. So again, this is just the idea, for the cats, again, represented schematically. Here's the zygote, maternally derived X, a paternally derived X, again, carrying, one of them carrying the big O gene or allele at the O gene, and the other one carrying the little o, so orange and black. At about that 1,000-cell stage, early development is finally when X inactivation occurs in each of those cells. It occurs in a totally random fashion, but it's roughly 50% of the cells will inactivate the maternally derived X, and about 50% of those cells will inactivate the paternally derived X. So you create a random mosaic or a random chimera. And then through subsequent development, producing a fully formed individual, you get the two colored cat, sectors of orange and sectors of black in a totally random pattern. Okay, so are there any examples of this in humans? Well, there are. They're not as immediately obvious as the coat color pattern in cats, but there is a gene on the X chromosome that causes this particular disorder, anhidrotic ectodermal dysplasia. And what it gives rise to, you have a normal functional allele for this gene that produces, a normal female, and then you are a normal human, and then you have a defective form, a defective allele for that particular gene as well, that gives rise to abnormalities. Okay, and one of the obvious abnormalities, what is affected by a defect in this gene are the formation of various ectodermally derived structures in the body. So things like nails, teeth, hair, skin, and so on. So one of the obvious defects is with the defective allele, the non-functional allele for this sex-linked gene is that you don't produce sweat glands. Okay, so what does that mean? Is so a male can only be hemizygous, right? So a male is either hemizygous normal or hemizygous defective, for the defective allele. And if he's hemizygous for that defective allele, again, you have all kinds of developmental abnormalities for all of these ectodermally derived structures, and you have uniformly an outer epidermal layer that is completely absent sweat glands. So a pretty, significant condition, a genetic condition. What about a female? So again, a female can be homozygous normal, okay, so perfectly normal for all of these ectodermally derived structures. She can be homozygous for the defective allele, so she would be just the same abnormal condition as a hemizygous male, so fairly severe condition. Or alternatively, she can be heterozygous. If heterozygous, then you're going to have sectors of the body that have normal ectodermally derived structures and other sectors of the body that don't. So again, she would be a mosaic or a chimera. So in other words, you could map the body. So in this depiction here, this female being a heterozygote, some sectors of the body would be absent, the sweat glands, and other portions of the body, the skin, would have a normal distribution of sweat glands throughout the epidermis. And if you compared multiple human females that are all heterozygous for this particular sex-linked gene, is that again the pattern of mosaicism or chimerism across the the epidermis of the body, again it's totally different, and again that's a reflection of the random nature of X inactivation that takes place at that early stage of development. Okay, so there's one other example of this. There's a bunch, as I've said before, the X chromosome of humans contains over 1,000 genes. So you can have defective alleles, and for these different genes, giving rise to alternative expressions for the traits conditioned by those genes, and therefore females, because this X inactivation takes place, can be mosaics or chimeras for whatever that particular trait is. So here's one other, significant example, I guess. This is a form of mental retardation that's referred to as Rett syndrome. And for many years, they couldn't explain why this particular form of mental retardation seemed to be occurring only in females and never in males. And so eventually what they were able to determine is that this condition is caused by a sex-linked recessive allele at a sex-linked gene. Okay, so again, what that means is that males can be either hemizygous normal, so if they're carrying the normal functional allele for the gene, they develop normally and they're born and perfectly healthy and normal. Alternatively, they could be hemizygous for the defective allele at this particular sex-linked gene. And what that means is that this gene is pleiotropic. Again, we talked about that concept previously, and that it can affect many different traits and characteristics. And actually, it causes developmental abnormalities to occur, during embryogenesis, during in utero development, and therefore most of the time, almost always, is that these males that are hemizygous for the defective allele are spontaneously aborted or stillborn. So you never see a live, viable male exhibiting the characteristics of Rett syndrome. So again, on the other side, the female can be homozygous normal. Okay, so perfectly normal. Normal in utero development, normal birth, and normal characteristics post-birth. Alternatively, she could be homozygous recessive, and again, in that state would achieve the same fate as a hemizygous defective male. In other words, developmental abnormalities occurring during in utero development, such that the condition results in a spontaneous abortion. However, if the female is heterozygous, again, what that means is that because of this X inactivation that takes place, is that you create a mosaic or a chimeric female. And depending on the nature of the mosaicism, that is then going to, so they can survive. They can survive in utero development, give rise to a live birth, and develop well beyond that point into adulthood. But the severity of the condition or the expression of Rett syndrome is going to depend on the nature of the mosaicism in that particular female. And again, remember, that is determined by the random inactivation of the X chromosome that takes place during early development. So there's a wide range of expressions for the different phenotypes that fall under, this, or fall within this condition that we call Rett syndrome. So you can read through the list down here of different characteristics, phenotypes, and behavioral characteristics that are affected in those that suffer from Rett syndrome. And again, then, the severity of the expression of these different characteristics is going to vary from female to female, depending on the particular nature of the mosaicism that occurs in that individual for, the pattern of X inactivation. Okay, so what I've described to you is, first of all, the general problem of a dosage imbalance that is created when you have sex chromosome differences between males and females of a particular species. So for autosomal genes, again, there's a normal dosage balance. The normal, as I said earlier, diploidy seems to be the normal state or the normal condition, 2 copies of everything. However, when you perturb that, again, certainly for an autosomal gene, this is going to cause developmental abnormalities and give rise to a genetic condition. Okay, so this dosage difference also occurs for the sex chromosomes. Again, as the examples we've been using is in humans, females 2 X chromosomes, males 1. So how do you balance things out? How do you equalize the dosage contributions or functionality of the genes on the X chromosomes? Actually, what you do is you shut down one of the X chromosomes in the females. So it's normal for humans to have one functional copy of an X-linked gene. Okay. So the mechanism for dosage compensation that I described, that's mammalian specific. Okay. So that doesn't happen in other organisms that have the same sex chromosome difference, males being XY and females being XX. That only occurs in mammals. So it's a very specific form of dosage compensation. Okay, but other organisms have the same problem. So, the one we've talked about is the fruit fly, Drosophila melanogaster. So again, in Drosophila melanogaster, females are XX and males are XY, but they don't dosage compensate through the same mechanism as we do or as mammals do. So there's in the cells of a female fruit fly, the somatic cells, you don't observe the bar body. Okay, the bar body does not form in that way. They have a different mechanism for dosage compensation because of this general problem, this dosage difference that exists between the two sexes for sex-linked genes. So what are I'm not going to go into the details as to what the fruit fly does. It's a much more complicated system of dosage compensation that involves other genes. So in other words, it's a genetically regulated mechanism for dosage compensation. But the end result is that you get equal expression of X-linked genes in both male and female fruit fly. So actually what happens through this genetic mechanism, this complex genetic mechanism, is that the one copy of the sex-linked gene in the males is twice as active as the two copies that are found in the female. In other words, the ability to express a gene product is upregulated. This single copy of the gene on the one X chromosome of the male can produce the same amount of gene product as the two copies that are present on each of the two X chromosomes in the female. So you get an upregulation of gene expression for these sex-linked genes in the male. That dosage compensates. You produce the same amount of gene product, whether you have one copy of the gene or whether you have two copies of the gene. So it's a different form of dosage compensation. So here's one other example. Here's another organism we haven't talked about yet. This is a C. elegans, or the nematode. And in this organism, you have two sexes, one of which is male and one of which actually is a hermaphrodite, has both male and female reproductive organs. So the hermaphrodites carry two X chromosomes, and the males only carry one. Okay, they don't have a second copy of a sex chromosome. They just have one X chromosome. So again, there's a dosage imbalance here, two copies of any sex-linked gene in the hermaphrodite, but only one in the male. So how do they dosage compensate? Well, again, it's a complex genetic mechanism in which actually instead of having upregulation in the male, you have downregulation in the hermaphrodite. So in other words, each of the two copies of these X-linked genes in the hermaphrodite are only half as active as the single copy of the sex-linked gene in the males. So there's a mechanism that makes these genes, the two copies of the genes, less active than they would normally be in a male. Okay. So again, the point of mentioning these two examples here is that dosage compensation is a general problem throughout nature for any organism that has a sex chromosome difference that differentiates male from female. Because you'll have different numbers of copies of sex-linked genes depending on whether you're a male or a female. So you have to dosage compensate. You have to equalize the contributions or the functionality of these genes between the sexes. So mammals do it one way. They just completely shut down one of the two X chromosomes. Drosophila does it another way through a genetic mechanism that allows for upregulation of gene expression. And the nematode uses a third mechanism for dosage compensation where actually they downregulate gene activity. Okay, so I'm going to stop there and we'll pick this up again in a future lecture.